I'm really glad to be here to present Xintela and our contribution to this exciting field of cell and gene therapy. I am the CEO and also the CSO of Xintela, and also the founder and the discoverer of the technology that is key in our development. Forward-looking statements here. So Xintela is a biopharma clinical stage company focusing on stem cell therapy, and we also have cancer therapy that is run by our subsidiary Xintela. I will talk about our stem cell therapy here today. In total, we are a team about 25 people based at Medical Village in Lund, in south of Sweden, and the bridge you see there will take you from the airport in Copenhagen in Denmark over to Lund in about 30-40 minutes. We're listed on NASDAQ First North in Stockholm since 2016, and the institutional investor Flerinvest is a major shareholder with circa 40% in the company. We have developed a superior stem cell product platform that we call XSTEM. We have two projects in clinical development in osteoarthritis and in venous leg ulcers. A proprietary stem cell marker, cell surface marker integrin alpha-10 beta-1, is a key in the development of our stem cell product, and I will tell you more about that soon. The XSTEM product is patent protected for all therapeutic uses for many years going forward. And we have also built our own GMP facility, so we produce our own products, have full control of all the manufacturing, which is a great asset. As you know, there are many different kind of stem cells in cell therapy, and we work with allogeneic adipose tissue-derived adult MSCs. And I'd just like to spend a little time on reminding you of the great advantage of using tissue-derived MSCs. And it's a list here. I'll just quickly go through it. No ethical concerns. They're easily accessible and easy to expand in culture and have really good proliferation potential. You can get clinical relevant number of cells that can be reached within a few population doublings, so there is a very low risk of genetic instability. They can home to the site of damage and differentiate into several cell lineages. They have the potential to treat a wide range of diseases without the need for pre-differentiation. They also have immune modulatory and anti-inflammatory capacity, and they're immune evasive and therefore also very suitable for allogeneic use. They're non-tumorigenic, and importantly, they have been extensively tested for safety in clinical studies. So we have developed XSTEM, an MSC product platform. And what is unique with XSTEM is the way that we select and quality assure a homogeneous preparation of stem cells. And in this selection process, we use this integrin alpha-10 beta-1 cell surface marker and antibodies binding to this marker. So we can actually solve this problem with heterogeneous MSC preparations that has been over the years a problem and still is a problem, to get consistent, homogeneous, high-quality MSC preparations. We have demonstrated that XM has functional and regulatory advantages over unselected cells. We can see improved differentiation, increased secretion of immune modulatory factors, improved homing to damaged cartilage, and importantly, as I already mentioned, also consistently high quality between donors. Just would like to show you one example on what difference this selection really makes. So if you look there at the homogeneous, the blue, if these are no two blue culture plates with a heterogeneous and with homogeneous XSTEM. If we take the selected stem cells and put them into pellets and differentiate them to chondrocyte, we see beautiful differentiation. If we take the other cells in this preparation, and remember now, all these cells in the heterogeneous preparation has these recommended markers, CD73, CD90, CD105. But if we take these cells and try to differentiate them to chondrocytes, they do not have the ability, as you see there, so they do not have the trillion differentiation capacity. So we have here a fantastic product with great opportunities. And we focus now on diseases, to treat diseases, huge unmet medical need. Our current focus is osteoarthritis and also difficult to heal leg ulcers. We've also performed preclinical studies in an animal, in a pig model for ARDS with beautiful data that is also now in line for clinical development. I also like to mention our equine product, XINT.ST. And I will tell you about some results from our preclinical studies in horses. So that is also a great opportunity to develop an effective treatment for horses with joint diseases. So we have decided to start this therapeutic journey with XSTEM in osteoarthritis. And an indication with really huge medical need. And as you see here, the lifetime risk of developing knee OA is 46%, and globally approximately 23% of all adults over 40 are estimated to have OA. And today we treat symptoms, pain, inflammation, and many of us end up with joint replacement. So, of course, there is a huge need for a disease-modifying osteoarthritis drug. Our preclinical studies, that I will tell you a little bit about later, really support a disease-modifying effect of XSTEM. We have, before this, we did this study that I will show you here. A few years ago, we did an OA study together with Lisa Fortier at Cornell University, demonstrating both safety and efficacy of our equine integrin alpha-10 selected MSCs in a post-traumatic OA model. This has been published in the American Journal of Sports Medicine. What I show you here is some more recent data that we did in collaboration with a group in Copenhagen, where we injected the selected MSCs into the carpal joint with post-traumatic OA, and then the horses were treated for 52 days and also compared to an untreated group. So what you see there in the upper left, so after 18 days after induction of OA, MSCs were injected, and we analyzed the lameness, had a lameness score here, so actually analyzed the pain, and we can see significant less lameness in the treated horses. And this was also very, we could also, nice correlation with a significant increase in modulatory factors. And you see there upper right, we could detect an increase after, you see the arrow there, after the MSC's injection, increased PD2, increased IL-10, increased IL-6, and so on. And also we had a very nice correlation to significant reduction in pathological changes. So to the lower panel there, you see less pathology or less degradation in the treated compared to the untreated. And I'd also like to show you some other data that I think is really beautiful. When we labeled XSTEM, our human product, with iron, so we actually can detect it by MRI, and we injected these cells into a rabbit joint with a cartilage damage. You see that to the far right, the far left. And then 4 days after the injection, we could see how the labeled MSCs really were homing to the damage. And when we analyzed this, and I also should say that these nanoparticles were also labeled with a fluorescence marker. So when we analyze and section this regenerated tissue, we can really see the presence of our stem cells there, and also see that they directly contributed to the regeneration. So we have a lot of understanding of the mechanism of action of XSTEM in OA patients, based on all of the massive preclinical studies that we have made. So we believe there will be homing to the degenerated cartilage, immune modulation, anti-inflammation with positive effect on the whole joint, including pain management, differentiation, integration and regeneration of the cartilage, and we expect to see disease-modifying effect. So of course, you understand we're now very excited to see the data from the ongoing clinical study that we have. It's a first-in-human phase 1/2 study in patients with systematic knee-OA. So this is a single intraarticular injection of XSTEM. It's a dose escalation study, 3 dose levels, 8 patients per dose. And we also have an option to the dose expansion up to 54 patients. So we do safety and preliminary efficacy assessment every 6 months, up to 18 months. The status now is that the first and second dose level has been completed and judged safe. And we are now prepared to start dosing the third and final dose. Right. So the second indication where we believe that XSTEM will really provide a great value for patients is difficult to heal venous leg ulcers, which is the most common leg ulcer. And it's difficult to heal when the wounds have not healed within 6 weeks. A lot of patients are suffering from this disease. It affects 4% of all individuals over 65, and there is no effective treatment available. Also in this case, we have performed really great large animal preclinical studies where we have demonstrated excellent wound healing capacity of XSTEM. We have started a clinical study in patients with VLU. This will be a randomized, placebo-controlled, single-blind study with 12 patients, single dose administered topically, and this will be added on to standard wound care. And then safety and preliminary efficacy assessed every 10 weeks, and the patient then will also be followed up to 6 months. And we are right now screening and recruiting patients to this study. So 23 we believe will be a very exciting year for Xintela with several value inflection milestones. We will have safety results from all dose levels in the OA study, and also expect to see assess early efficacy signals. In the VLU study, we expect to have during 23 safety and efficacy study, and we also expect to be able to do some great advancement in development of the equine product XINT.ST for treatment of horses. And we definitely look for partners and investors also to advance the further development of this great product, XSTEM. Our goal is to partner XSTEM for venous leg ulcer and osteoarthritis after safety and preliminary efficacy readouts. We also are looking for partners that will, help us to advance the development of XSTEM also into other indications that we are not currently running ourselves, and including ARDS. And from the data that I also presented from the horse studies, we believe XSTEM will also be a fantastic product for horses. So we're also looking here for a partner to do further development and commercialization of XSTEM for treatment of joint diseases in horses. And with that, I end my presentation, and we're happy to answer questions after here. I also have 2 people from Xintela, head of manufacturing and head of CMC, are also here present at the meeting. So please approach us reach us if you have some questions. Thank you.